Welcome to this video describing our recent paper in JCI Insight, in which we describe a novel classification of thick ascending limb cell types that enables the fine control of electrolyte excretion. I am Dr. David Ellison, Professor of Medicine at Oregon Health and Science University. The paper was the result of an international collaboration between Sebastian Bachman's group in Berlin and our group at Oregon Health and Science University, as well as groups in Kiel, Germany, and New York. Professor Bachman, please introduce yourself, provide some brief background, and describe your initial findings.
My name is Sebastian Bachman. I'm an anatomist of the Charité Hospital in Berlin, Germany, whose work is specialized in experimental nephrology with emphasis on histopathology and cell biology of the nephron. One of my projects has a focus on the loop of Henle, and here particularly on the thick ascending limb, or TAL, of the kidney. As shown on the slide, the TAL courses through the inner and outer stripes of the outer medulla and into the cortex, where it includes the macula densa at the spot where it touches its own glomerulus. This slide shows important TAL functions, which include sodium chloride reabsorption, urine dilution, countercurrent multiplication, calcium and magnesium reabsorption, and hormonal regulation. Of note, this region of the nephron is also the target of loop diuretics, including furosemide, and a site in which genetic mutations cause human disease. Based on the pioneering work of Moberg and Rainer Greger, thick ascending limb cells have generally been conceived as comprising a single type. As shown, transport is energized by the sodium pump at the basolateral membrane. Sodium, potassium, and chloride enter across the apical membrane via the electroneutral 3-ion cotransporter, the target of loop diuretics. Chloride leaves through a channel at the basolateral membrane, and potassium leaves through a channel at the apical membrane, creating a lumen-positive voltage. This voltage drives paracellular sodium, calcium, and magnesium reabsorption. There have been hints in the literature, however, that not all TAL cells were the same. As shown on the left, early studies in Amphioma and hamster TAL segments showed that the apical and basolateral potassium conductance follows two patterns, with basolateral conductance on the x-axis and apical conductance on the y-axis. More recent studies, diagrammed on the right, showed that monovalent and divalent paracellular transport is mediated by two types of claudin molecules, which are expressed in a mosaic pattern. Using immunocytochemical techniques, we found that the apical potassium channel ROMK is not expressed in all TAL cells. As shown here by the graph at the bottom right, quantitative analysis showed that from 25 to 42% of TAL cells do not express the ROMK at the apical membrane, and the percentage of positive cells varies by kidney zone. When we analyzed the expression of the basolateral potassium channel Cur 4.1 in the same way, we found it only in cells that don't express ROMK at their apical membrane. Interestingly, the calcium sensing receptor, CASR, is also expressed predominantly by ROMK negative Cur 4.1 positive cells. When we examined the expression of claudins, we could confirm prior work showing mosaic expression of claudins 10 and 16, but given our findings about potassium channels, we extended these observations to show that cells ringed by claudin 16 express basolateral Cur 4.1, but not apical ROMK, whereas cells ringed by claudin 10 were ROMK positive.
The results just described recall earlier work suggesting that TAL cells possess two types of potassium conductance. To test this in mice in collaboration with Wenli Wang at New York Medical College, we used the ROM-K selective inhibitor TPNQ. As shown on the left, comparing the blue versus the red dots, only about half of cells exhibit measurable ROM-K activity. Then, to address sidedness, we collaborated with the Blake Laboratory in Kiel, which used perfused tubules to change basolateral potassium concentration. As shown on the right, comparing red versus blue dots, only some cells responded with a change in voltage when basolateral potassium concentration was increased.
Hello, I'm Jessica Bahena, postdoctoral fellow at Oregon Health and Science University. To explore whether the cell type diversity we detected using immunocytochemistry and electrophysiology corresponding to transcriptional heterogeneity, we wanted to enrich for tall cells and perform RNA sequencing. We therefore used the isolation of nuclei tagged in specific cell types, or INTAC method. We crossed NKCC2 Cre mice with intact reporter mice, selectively labeling NKCC2 expressing nuclei with green fluorescent protein. After dissecting cortex and medulla, GFP positive nuclei were sorted using fluorescent activated nuclei sorting, and then subjected to single nuclei RNA sequencing. On base clustering analysis of the enriched tall dataset, identified distinct cell types that were consistent with the functional and morphological findings just described. One cell type expressed the monovalent cation selective for Claudin 10, but lacks KCNJ10, the basolateral potassium channel Kirchhoff.1. We had labeled these cells alpha cells. A second cell type expresses the divalent cation selective for Claudin 16, together with KCNJ10. We have labeled them beta cells. A third population co-expressed Claudin 10 and KCNJ10 and is restricted to the inner striate of the outer medulla. We have labeled them as gamma cells. In addition to these main tall subtypes, we identify macula densa cells and a small cluster of proliferating cells. To further explore functional specialization, we analyzed differentially expressed genes across tall subtypes. Tall alpha and tall gamma cells, which express the monovalent cation selective for Claudin 10, show increased expression of sodium handling regulators, including Wim4 SPAC and the vasopressin receptor. In contrast, tall beta cells, characterized by the expression of the divalent cation for Claudin 16 and the basolateral potassium channel KCNJ10, display elevated expression of genes involved in calcium and magnesium homeostasis, such as the calcium sensing receptor, the PTH receptor, and the vitamin D receptor.
To summarize, by combining immunocytochemistry and transcriptomics, we identified 3 distinct cell types along the TAL that we label as alpha, beta, and gamma cells. We suggest that it is only alpha cells that generate the transepithelial voltage and mediate the majority of sodium reabsorption, both through the cell and paracellularly. In contrast, beta cells do not generate a voltage, but instead use the voltage generated by alpha cells to drive divalent transport. Gamma cells are only found in the inner aspect of the outer medulla. We speculate that these hybrid cells reabsorb not only sodium, but also potassium. If you're interested in learning more about these results, please take a look at our paper, and thanks for listening.